genetic imprinting. Printing. Print means printing, typing. What is that? Printing. Living markings. So leaving some marking. So genetic imprinting. Differential manifestation of gene. So differential manifestation of gene is known as genetic imprinting. That means, very simple, genetic imprinting is a concept of behavioral genetics. Behavioral genetics. See, if we receive male, female, that means father, mother. Father is tall, mother is dwarf. What about the offsprings? If tall is the dominant, definitely all children will get tallness. Is it? This is the normal rule. No, if father is dwarf, mother is tall. In that case, reverse also, result is same. I mean, here the gender of parent is not the criteria. But this rule is not a universal rule. See, if father is tall, mother is dwarf, children are tall. Even in reciprocal crossing also, result is same. That is known as law of equivalence, law of equivalence. That means, if father tall, mother dwarf, or father dwarf, mother tall, in any case, children are tall. That rule is known as law of equivalency or equivalence. Yeah. This rule is not a universal rule. Sometimes the gender of parent decides the manifestation. When gender of parent decides the manifestation, then that comes under imprinting. That comes under imprinting. Say for example, in autosomal case, both male and female are equally, is it? Equally prominent in the process of inheritance, is it? Yeah. If we receive same trait from the father, that means male to father to either son or daughter, definitely both, is it? If father is tall, both son and daughter is tall. Again, this daughter tall, her husband also tall, then even her son and daughter also tall. If that is autosomal case. But in some diseases, this case is totally reversed. Say for example, Huntington disease. Huntington disease is an autosomal disease, is it? So neuromuscular degradation, is it? Gradual degradation of the neural cells that leads to loss of memory, amnesia, and even number of neural problems. Finally, that leads to death of an individual, is it? Almost the symptoms arise after 40 years. Yeah, in between 40 to 60, of course. See, if father is a Huntington patient, Huntington disease. Yeah. If father is Huntington, then definitely both son and daughter will receive that disease. If the person is homozygotic, is it? Definitely both will receive, but here symptoms are very negligible. At the same time, even delayed. Symptoms are delayed and symptoms are very low. Right. Father no origin. If father is the sufferer, both son and daughter receive very early and severe. Then from daughter to, that means mother to, if they receive, then here the symptoms are delayed and the symptoms are very negligible at low rate. Yeah. Again, from son to, that means male to, if children receive, again the symptoms are either son or daughter. Again symptoms are early and severe. If they receive from the mother, then again delayed and low. That means this manifestation of the symptoms is depend upon the origin of the gene. Origin of the gene, particularly on the basis of the gender of parent. Such a differential manifestation based on the gender of the parent, differential manifestation based on the gender of the parent is known as imprinting. Yeah. Differential manifestation of gene based on the gender of the parent. Differential manifestation of D gene based on the gender of parent is known as imprinting. Example, Huntington disease. Autosomal dominant, of course. Autosomal dominant. Symptoms arise after 40 years, is it? Yeah. So, in this case, if father is a sufferer, if he is homozygotic, 100% sons and daughters will receive the gene, but the manifestation is very delayed. Manifestation is very early. At the same time, very severe also. Very severe. Almost within 40 years, definitely the symptoms arise. Within 40, symptoms arise. Yeah. Now, from daughter to, that means mother to the children, both sons and daughters received the gene. Then the symptoms are delayed, maybe almost at the age of 60 plus. At the same time, the symptoms are not severe, symptoms are very mild, very low rate. Yeah. At the same time again, if they receive next generation from the father, again early and severe. If they receive from the mother, then again delayed and mild. Yeah. Such a gender-based differential manifestation. Is known as imprinting. I hope it's clear. Then characters of imprinting. Wait. Characters of imprinting. So, characters of imprinting. So, these are all gender based. First character. These are all gender based manifestations. Then, its magnitude is differ based on the gender of the parent. Its magnitude is differential magnitude based on the gender of origin. So it may be enhancing or retarding effect, or the effect may be enhancing or retarding. The effect may be enhancing or retarding. Yes. Yeah. So sometimes the symptoms may be turned off. That means totally absent sometimes, no question of arising. Then again, the symptoms are erasable. That means sometimes they are going to erase generations together. No way manifestation, silent, erasable. Then next, it is not a permanent phenomena. It is not a permanent phenomena, then it is not a rule. Then it is species specific. It differs from species to species again. It is species specific. Differ from one species to the another. Again, it is gender specific. Differ from one gender to the other based on its origin. So, like that, this imprinting, very simple concept, nothing. What are the reasons? Nothing, very simple. Yeah. See, in women, that means in mother, woman, yeah. So the ova will form when this woman is in her mother womb. That means the age of ova is equal to the age of the woman. That means the ova will form almost in her mother's womb, after birth. No question of formation of the ova, just some changes and releasing, that's it. Almost all chromosomal separation takes place at the time of birth itself, before birth itself, at the time of embryonic development itself. Yeah. So, almost in anaphase, the chromosomes are going to arrest, finish. After anaphase, telophase, that's it. That last telophase only takes place in that woman. Remaining everything happened in her mother womb only. That means the ova age of the ova is equal to the age of the woman. So, a prolonged preservation, almost from embryonic time to the ova chromosomes are fixed and preserved. Then after puberty, releasing of sex hormones and getting stimulus and releasing 1 month after 1 month after 1 month after, is it 28 days once, one release, next another one, next another one, next another one, something like that. So, due to prolonged preservation, so high methylation. That means these methyl groups, CH3, are going to accumulate, continuously going to accumulate. Due to preservation and accumulation of methyl groups, the chromosomes will become almost stuff. That's why at the time of separation, they are going to break out, or they are almost clubbed together. That's why in women only chromosomal aberrational chances are very high. That's why in women, chromosomal aberrations arise only in the woman. Reason is this long-term preservation only. Due to this, spindle fibers also will break out. Chromosomes also will break out when they are going to separate. So automatically, number of defects from mother side. At the same time, even though they are equally separated, due to high methylation, manifestation is not possible. Reason is, almost we have two sets of chromosomes. One set we receive from the father, another set we receive from the mother. Always, what are the active genes? Father genes. Father genes are always active. Mother genes are always inactive. Reason is methylation, not only X chromosomes. In women, even though they have two X chromosomes, one only functional, other one is not functional. Is it? Not only X chromosome, even autosomes also same to same. So, all chromosomes are not functional. One set only functional. That's it. Yeah. But in X chromosome case, absolutely due to methylation, one is unfunctional, non-functional, another is a functional. So this is the reason for imprinting. Yeah. This is genetic imprints in human disease. Just 5 to 10 minutes topic. Yeah. Then genetic counseling. Yeah. So genetic counseling and genetic screening both are of course linked with one another. Yeah. Genetic counseling. What is this genetic counseling? We discussed already in pedigree analysis. Is it? In pedigree analysis, so the researcher, the geneticist, is it? After collecting the family history by using some specific symbols, internationally accepted, is it? The researcher construct pedigree analysis. Why he construct the pedigree analysis? To understand the process of inheritance and to understand the risk factor. Is it? By estimating risk factor, this geneticist suggests take such a something, precautionary measures, how to prevent it. What about the risk factor? How much risk is there? If homozygotic dominant, 100% autosomal. Is it? If autosomal heterozygotic dominant, 50%. On the basis of that, doctors will suggest. So these are all end topics, just time passing 10 to 15 minutes. Yeah. So genetic counseling is also a pedigree analysis. So on the basis of that, yes, he analyze and explain how much risk is there. So something like that, it goes on. What are the notes? Genetic counseling.
Why is it that only in imprinting cases is happening? Because we have discussed a lot of aberrations, right? So we
Chromosomal aberrations also due to this one only, imprinting also due to this one only. Is it? These are all, that means this methylation is a major reason for all, many. Genetic counseling. Genetic counseling. So whenever a family or an individual is suffering with a known or unknown disease, then they consult to a geneticist or a medical professional. So that is known as genetic counseling. Here the person who is seeking advice, genetical advice, is known as counselee. Who is giving advice, he is known as counselor. Yeah. So the counselor must be a well-trained geneticist or definitely a medical practitioner. At the same time, the counselor should have proper knowledge related to the human diseases, genetics, law, even social rules. Reason is, ethical point of view, number of problems arise. Doctors can't display the case in front of the others. In society we have our prestige. That person has some disease, this person has some disease, their family is suffering. What about the prestige in the society? So the counselor should have not only medical knowledge, genetical knowledge, he should have social knowledge, social rules and regulations, law, and many. Yeah. So definitely it comes under something like that. So here the counselor has only right to advise. He has right to advise. He can't demand, boy, you are getting number of diseases, your wife also has disease, your children will get disease, abortion karo simply. What right you have? If my son is sufferer, I am ready to bear it. I have to take the decision. Who are you to command me? Doctor's rule, doctor position is only advising, explain the risk and remedial measures. Take this is the measurement, then you will get something. Reason is, this is the probability. This is the probability. This person is suffering with Huntington disease. Wife also suffering with Huntington disease. How can you say these two are going to fertilize? Why can't these two fertilize? Is it? Chance is 50%. Yeah. Is it? You explain the risk. Explain the risk. Yeah. That's it. Of course, here, these two Huntington will come. Again, these two Huntington will come. These two Huntington will come. 75% risk is there. 25% in this one is there. Yeah. But each time probability. So, there is a chance to fertilize these two at the same time. Equal chance for these two. Yeah. 75-25 not applicable here. 50-50 only applicable here. That's it. That's it. Chances are there. So, explain that chance. Brother, you have a lot of risk. Then finally, leave it to the sufferer, whatever he may be. So, even though he is ready to get defective child, what right we have to prevent that? Is it? Yes. So, the debate goes on like that. Yeah. So, the only duty of the genetic counselor, that is constructing the pedigree, analyzing the risk factor and explaining and also suggesting some preventive measures. That's it. He has no right to pressurize or the abortion. The final decision goes to counselor, the counselee. So, the debate goes on like that. Study now already. That's it, end of the matter. Okay. Write it then. Genetic counseling. Genetic counseling is a very popular concept in eugenics. Genetic counseling is a popular or significant concept in eugenics. What is this eugenics? Welfare genetics. Is it? Welfare genetics, the true genetics, whose aim is to eliminate undesirable genes and to establish healthy society. So, genetic counseling is the most significant concept in eugenics, whose aim is to eliminate undesirable genes and to establish healthy society. In present times, it is playing a prominent role due to increasing of genetical problems in the society. Then it is a case study approach. It is a case study approach. Whenever a person or a family is suffering from a particular or unknown genetic disease, whenever a person or a family is suffering from a particular or unknown disease, they need genetic counseling. Genetic counseling is useful to understand the risk factor and to prevent undesirable genes. Next, it is a positive approach in eugenics. It is a positive approach in eugenics. It is an advisory approach. It is also useful to take proper remedial measures. The person who is taking advice, or who is seeking advice is known as counselee or consultant. COUNSELEE. Counselee or consultant. The person who is giving advice is known as counselor. It includes, that means genetic counseling includes pedigree analysis, calculating the risk factor, remedial advice, and treatment. The counselor should have genetical, medical, social, legal, and psychological knowledge. A genetic counselor must be a medical doctor or a well-trained professional of human genetics. Ideally, it is a case study approach. In this, the researcher should analyze the case. The researcher should analyze the case in all the dimensions. After studying the case by using pedigree analysis, biochemical techniques, karyotype, a genetic counselor is supposed to give all sorts of information and leave the choice to the counselee. The counselor has no right to compel or to impose anything. In extreme conditions only, the counselor can recommend abortions. While disclosing the information, the counselor should take a lot of care so that the counselee should not psychologically upset.
Sir, while disclosing the information, the counselor should take a lot of
care so that the counselee is not psychologically upset. The advice must be secret, should not disclose openly. This is about genetic counseling. Nothing here, nothing. Yeah. Then genetic screening. Genetic screening. Screening means preventing. This genetic screening also part of genetic counseling originally. Yeah. So in genetic screening also the researcher should use all the techniques. What are the techniques? DNA analyzing techniques, pedigree analysis, then karyotype analysis. After analyzing all these, and to analyze the genes and chromosomes, so the researcher can use different enzymes and techniques. What are the enzymes? Restriction, endonuclease, and ligase. Yeah. This restriction endonuclease is useful to cut and separate the gene. This ligase is useful to paste the genes. Then here we can use some techniques. What are that? Of course, these are all pedigree technique. PCR, polymerase chain reaction, gel electrophoresis, then blotting techniques, in blotting techniques, southern blotting, northern blotting, and western blotting. So this blotting techniques, southern blotting technique is useful to confirm the DNA, and northern blotting technique is useful to confirm the RNA. The western blotting technique is useful to confirm antigen antibody. At the same time, each and every biochemical substance has its molecular weight and some charge. On the basis of molecular weight and charge, we can identify all biochemical substances. So any abnormal substance, if it is present, we can understand by using this gel electrophoresis. At the same time, this pedigree analysis, it depicts the history of the disease. At the same time, PCR, polymerase chain reaction, this technique is useful to produce abundant number of similar copies, true copies, Xerox copies. So here we are using number of techniques and enzymes. At the same time, ultrasound, techniques are blood sampling, urine sampling, so blood and urine analysis, and finally analyzing of DNA analysis, and even karyotype analysis. Then after analyzing all this, collecting the blood or serum or urine, analyzing that, then collecting the DNA and analyzing the DNA and constructing the karyotype of a particular person, then so the genetic counselor suggests what type of risk factor is there, how to prevent the disease, whom to marry, whom not to marry. If the person has heterozygosity, then definitely doctor can understand what type of problem this person has, something. At the same time, the screening takes place in 3 stages. The first one, prenatal. That means before birth itself. Then in second stage, neonatal, immediately after birth. In third stage, that is adult, in adult stage. So this genetic screening takes place in 3 stages: prenatal, before birth. Here we use amniocentesis method. What is this amniocentesis method? Around the baby, some fluid is there. Is it? Injecting needle and collecting that fluid, amniotic fluid, and analyzing DNA and karyotype analysis. Is it? That is known as amniocentesis. Otherwise, chorionic villi, sampling. Chorionic villi sampling test. Otherwise, they collect blood and urine of the mother and they can analyze the sampling. Then, if they identified any abnormal substances in the blood or urine of the mother, on the basis of that, they can understand what type of problems the baby has. So, in this prenatal, definitely we can understand this biochemical disorders, at the same time chromosomal aberrations, in prenatal, Down syndrome, Edwards syndrome, Patau syndrome, Cry-Duchar syndrome. Is it? So, if doctors identify these aberrations in karyotype, then they can suggest. So, some problems are there. If you want to go for abortion, go for. Otherwise, that decision left to the sufferers only. That means the counselee only, not the counselor. Then in neonatal, immediately after birth, so again they collect the blood sample or urine sample of the baby itself, then they analyze the DNA or karyotype. Then here, if they understand the conditional lethal, then we can save the child. If phenylketonuria diagnosed, then what is the problem in phenylketonuria? So, if they consume phenylalanine diet, then only the problem creates. If they consume phenylalanine free diet, nothing problem will arise. Is it? At the same time, G6PD, glucose 6-phosphate dehydrogenase. This is also a conditional lethal. Even though this deficiency gene is there, the individual survives. Is it? Normally. But if they consume fevabine, is it? If they consume fevabine, then only that leads to death, favism. If we avoid that, that individual successfully survive. That means in prenatal one can diagnose the biochemical disorders and chromosomal aberrations. In neonatal, is it the technicians or geneticists can understand, can diagnose the conditional lethal like phenylketonuria, G6PD deficiency in adult. So, if they diagnose these heterozygotes, say for example, the person has normal hemoglobin A, another hemoglobin S, heterozygotes. If they understand the position of the genes, that means heterozygotes, then they advise whom to marry. If this person marry normal woman, whatever, normal person, then no child will suffer. Is it? All children are normal. If that person marry another heterozygote, then only problem arise. 25%. Percent is not the here. These two may fertilize, otherwise these two may fertilize. So 50-50 chances only in general, but in specific 25%. In specific, in random fertilization only. Yeah. Whatever, if the heterozygote marry another heterozygote only, problem arise. If a heterozygote marry a normal person, then automatically he can escape from the risk. No child will get the disease. Something like that. Is it? So the geneticists or genetic counselors suggest whom to marry, whom not to marry. That means genetic screening and counseling almost linked. Without screening, how genetic counselor advise? Is it? Not possible. So genetic screening takes place in 3 stages: prenatal, neonatal and adult stage. In prenatal, they use amniocentesis, coronary villi sampling, blood and urine sampling, blood and urine of the mother. Then they analyze DNA and karyotype. So here the geneticist can understand biochemical and problems in neonatal immediately birth. So the child is available, then they collect either blood or urine of the child, then they analyze the DNA and karyotype. Then if the technicians diagnose this conditional lethal, then they can protect the child. Then in adult, if they diagnose heterozygotes, then they can suggest whom to marry, whom not to marry. Something like that. This genetic screening, it goes on. In DNA analysis and construction of the karyotype, in karyotype we have seen something. So what type of enzymes they are using? Is it what type of techniques they are using? Is it? This is something. So pedigree. So it is useful to extract the history and the risk factor. PCR, polymerase chain reaction. It is useful to estimate produce a number of copies. Gel electrophoresis. It is useful to understand the biochemical substances and the nature of that. Then these blotting techniques are useful to diagnose DNA, RNA, antigen and antibodies. This is southern blotting, this is northern blotting, this is the western blotting. Then ultrasound, blood and urine analysis, something. These are all last topics actually. After completion of the syllabus we discuss. Yes. Hmm? 15 to 20 weeks, not in between. See, if baby, after developing of the baby, if anything procedural problems arise, that leads to abortion also. 1 to 2% risk is there of abortion. So generally, as per the conditions, so they go for first mother serum test, blood test, urine test. That is the first diagnosing. In that, if they are unable to diagnose any Then chorionic villi sampling they prefer. In chorionic villi sampling also almost in between 9 to 14 weeks only. After that is also not possible. The chorionic tissue is going to mix with the mother tissue. Then wrong diagnosis only. Even in that also chorionic villi extracting is very problematic. When you inject needle, the needle passes through mother's tissue. Then automatically mother tissue also will enter into the needle. So, in any method you can't diagnose properly. Yes, that is true. At the same time, of course, chorionic villi sampling we do 9 to 14 weeks. Then after 15th week, no method is working. Then amniocentesis is inevitable. Amniocentesis is 100% success, but 1 to 2% risk also there. Definitely in a something leads to abortion. That leads to abortion. Maybe mother tissue when injecting needle definitely penetrate through the mother tissue. Then mother tissue enter into the amniotic fluid. Number of infections. So no test is absolute, but of course. Even a number of injection if you use, definitely there is a problem of HIV also, contaminated needles if you use. 90% in India, the dentists and normal who is that what is it, doctors? No, Ayurvedic homeo, no, this untrained some professional rural area. Doctors are genetical, that means clinic practitioners, medical practitioners in rural area. Compounder, not that. So something, they know some medical knowledge. So they have certificate also. They can definitely use RMP. Registered Medical Practitioner, RMPs. This RMP, through RMP and dentist, HIV is spreading like anything. Dentists also very danger. They use maximum machinery to clean the teeth and something. That machinery even after sterilization also. No HIV you can't kill. Radioactive sterilization only is very costly. That's why simply they use the hot water, boiled water, something. Then simple sterilization. Then using the same equipments for the others. Then automatically if one patient in somewhere, end the matter. Yeah, of course, Gangaram is very popular, no? We can believe it. That is a problem in general hospitals. This medical practitioners, RMPs, registered medical practitioners, they use same needle. Even they never change the needle also. Simply in hot water dipping, cleaning. Yeah, simple for all same needle. Due to them, HIV is spreading like anything. They think we are cleaning. अरे भाई cleaning में HIV नहीं जाएगा. Even radioactive cleaning also, if not properly done. So, the mean. But what we can do, this is the poorest poor country. It is not developing country. It is underdeveloped country in real sense. It is the richest country. People are very poor. Richest country, people are very poor. Reason is the money is concentrated only on not less than 5% population. Just 5% population has more than 85% income of the country. Remaining 95% public has just 15% revenue only. This is the survey I am saying. Yes. Only 1% population has almost more than 50% revenue in their hands. Such a richest people are there. In India, there is the lowest in the world. As per the budget rules, as per the rules of Western originally, 10 to 15% of the revenue must be allotted to the health. But in India, that is not even 1%, 1 to 2%, that's it. 1 to 2% only. That's it. 1 to 2% only from the beginning. Yeah. So, at least 10 to 15% budget should allot to the public health. There is no public care here. Education also say 1 to 2% only. Even as per Kotaria Commission, at least 3% should invest. At least Kotaria Commission recommendation, if you follow, they follow. Minimum 3%, it recommended. But at least that also we are not following. Then what to do? UP is the worst state in India. UP, Bihar. They won't care about the public. Humans are living just like animal, not even an animal. At least in jungle, animal has absolute independence. But in Bihar, of course, after anything. Throughout India, that's there, brother. Throughout India, all government doctors managing their own clinics. If any patient came there, simply they recommend go there. You come there. Directly they give prescription, not writing, printed prescription they are giving. The public, the clinic name, address, phone number, everything. Even in Gangaram also they are giving. In Gangaram also they are giving directly their public, the original prescription they give. In Gangaram also they are giving. In AIMS also they are giving. AIMS, the general card is there. In general card they were never used, but if you consult a specific doctor, he give. I went no for this problem. I went AIMS, I went Gangaram, I went. Whatever the case, where we entered, why are you inviting to your public private clinic? Why are you inviting? Where? No, not recommended. But simply they are given injection. That's it. That's there. I know, but not for me. Only I have taken injection only. The Botox injection only I have taken. Even where you have to purchase the medical also they recommend. Where you have to purchase the medical, where you have to, that means the diagnosing tests, that also they recommend. Go there only, go take medical this only. They want percentages and all. What we do? That to common man, automatically he believe, why he recommended no go there only. So we can't. Okay, whatever. That is not our topic. Yeah. So genetic counseling, screening. Yeah. What is genetic screening? You can know? Yeah. Shall we start sub topic? Yeah. Yeah. See, I am repeating again. Genetic screening. What is genetic screening? Genetic screening also a medical. Write it otherwise. Genetic screening is a medical experimental technique. Genetic screening is a medical experimental technique useful to extract and analyze. Chromosomes. Useful to extract and analyze the chromosomes to prevent undesirable to prevent undesirable diseases. Genetic screening is always linked with genetic counseling. It plays a prominent role in eugenics. In genetic screening, technicians use number of enzymes and techniques to analyze the chromosomes and genes. Among enzymes, restriction endonuclease and ligase are the prominent. Among the techniques, PCR, gel electrophoresis, blotting techniques, ultrasound, blood and urine analysis, DNA analysis and karyotype analysis are the prominent. Even pedigree analysis also play prominent role. PCR, polymerase chain reaction. PCR, polymerase chain reaction. It is useful to produce multiple copies of DNA. Next, gel electrophoresis. Gel electrophoresis. To produce multiple copies of DNA. PCR, polymerase chain reaction. Gel electrophoresis is useful to analyze biochemical molecules on the basis of molecular weight and charge. Blotting techniques. In blotting techniques, southern blotting is useful to confirm DNA. Northern blotting is useful to confirm RNA. Western blotting is useful to confirm antigen and antibodies. Then pedigree analysis. Pedigree analysis is useful to construct family tree. These are all we know. Next, genetic screening takes place in 3 stages. First one, prenatal, 3 stages. First one, prenatal stage. It is an intrauterine analyzing. It is an intrauterine analysis. It is useful to analyze chromosomes and biochemical substances. In this method, technicians can diagnose biochemical and chromosomal disorders. Examples: biochemical and chromosomal disorders. Examples: Down syndrome, Edward syndrome, Patau, Klinefelter. Some example I am giving, that's it. Yeah. Next, neonatal. In neonatal diagnosis, technicians use blood and urine samples of the baby. Blood and urine samples of the baby. It is useful to detect conditional lethal diseases. Example: phenylketonuria and G6PD. This method is very useful to prevent the conditional diseases. This method is very useful to control conditional lethal diseases. Next, adult diagnosis. This method is useful to analyze heterozygotes. If heterozygotes are diagnosed, the genetic counselor can suggest whom to marry, whom to marry and the risk factor. The genetic counselor can suggest the risk factors and whom to marry. Yeah. Example: sickle cell anemia. Sickle cell anemia, hemophilia. Is it? These are all heterozygotic, no? Something. Yeah. Genetic counseling, genetic screening, imprinting. Yeah. Then DNA profiling is nothing but the DNA technology, shall we see later on. Gene mapping also comes under, shall we see, genome study. Genome study. Yeah. Already we know the genome study. Chromosomes, chromosomal classification, chromosomal analysis. Is it? So just 5% DNA only functional, remaining 95% is a junk one. So if you read totally that one, that itself genome. Yeah. So genome, the first time the term genome was proposed by Hans Winkler. H-A-N-S, Hans, W-I-N-K-L-E-R. Hans Winkler in 1920 suggested the term genome. This genome term derived from gene plus chromosome. In gene, first 3 letters gen, in chromosome, last 3 letters om. Yeah. genome, Genome, gene plus chromosome, genome, Hans Winkler, 1920. Then what is genome? Very simple. The total genetic constitution of an individual is known as genome. Write it. Total genetic constitution of an individual is known as genome. That means total hereditary information. Total hereditary information encoded in DNA. Total hereditary information encoded in DNA. That means total genetic constitution of individual. That means again total hereditary information encoded in DNA is known as genome. I hope it's clear. Is it? Yeah. So genome includes all chromosomes and mitochondrial DNA. Mitochondrial also DNA, no? Separate DNA, that one. Yeah. Genome includes all chromosomes and mitochondrial DNA. Yes. That means genome includes again both functional and non-functional DNA. So generally among DNA only 5% is functional. Generally among chromosomes, is it only 5% is functional, known as genes. Remaining 95% is non-functional, known as junk. Junk, J-U-N-K, junk. Yeah. Then write all that chromosomes, chromosomal analysis, their structure, is it? Yeah. Yeah. Then DNA structure, you know DNA structure. Yeah. So DNA includes the sugar molecule, phosphate molecule, nitrogenous bases. Sugar molecule, what is the sugar molecule? Deoxyribose, sugar. Yeah, the phosphate, what is that phosphate? Phosphoric acid. Nitrogen bases, how many types? No, types. Purines and pyrimidines, 2 types. Purines are, what are that? AZ, adenine, guanine. Pyrimidines are TC, CT or TC, thymine, cytosine, is it? So one purine matches with pyrimidine, is it? AZ one pair, AT one pair, GC another pair, in between AT 2 hydrogen bonds, in between GC 3 hydrogen bonds. You have to write this total DNA structure, total number of chromosomes, chromosomal analysis, is it? You have to write the total. If you write the total, that is known as genome. That means total genetic constitution of an individual, that is known as genome. I hope it's clear, is it? Finally, after writing all that, then again Human Genome Project, write this, is it? Human Genome Project. Human Genome Project started in 1990 and ended in 2003. Human Genome Project 1992-2003. So, Human Genome Project is an international project. Human Genome Project is an international project. Determining of all nitrogen base pairs and their sequences is a aim, and its aim is determining of all nitrogen base pairs and their sequences. Its aim is determining of all nitrogen base pairs and their sequences. That itself is a genetic information, no? Is it? Nitrogen base sequence only, no genetic information? Yes. And finally, preparing gene mappings, gene maps, and finally preparing gene maps. This is the aim. Is it? Yes. Then, according to Human Genome Project, in human 3.2. According to Human Genome Project, 3.2 billion base pairs are there. 3.2 billion base pairs are there. Yeah. This is equal to 3.2 GB, gigabytes. Is it? 3.2 GB, gigabytes. Yeah. Then human genes and ape genes are 99.9% same. Human and ape genes are 99.9% are similar, same. At the same time, man and pig genes 98% similar. Then all humans genome is similar. All human genome is similar. Only 0.05% only variable. All human genome is similar. Only 0.05% only variable. Human and animal genome also just 5% only variable. Human and animal genome also just 5% only variable. No, 5%. 0.05 in human. Not even 1. 0.05. Is it? In human. But animal and man compare 5%. Maximum 5% only variation is there. In some, see the apes, 99.9% similar. In pigs, 98% similar. Pig and human. Is it? Total animal. If you consider animal and human, maximum 5% only variable. This only the confirmed. Yeah. This is about genome. That means genome already we discussed. That's it. After completion of the total syllabus, 1 minute we can analyze that. Yeah. So, DNA technology shall we discuss tomorrow. Which one? 9 point? No, don't worry, I will give. Tomorrow class is there, then after tomorrow class is there. Yeah, 30th evening, your class up to 6 the class is there.